Amy.
Hi there. Is anybody able to hear me?
Yes, sir.
Okay, good. Hi, Jamie.
Hello.
Okay, so we're a little early, but what I have here, we're going to screen share, and we're going to look at some of these images, and then later on we're going to talk. We'll go over the our assignments that we actually have. So what I will do is to see who's trying to get in and let them in. And so hopefully we get at least a good 12 people here because I want to go over a lot of material. Okay. So let's get that down. All right. So it's just a minute before class time would start. We have an eyeball on the screen. I can see it. Can anybody else see it?
Yes.
All right, good. All right, that'll help. So if we have time, we're going to review all the images for all the 3 labs, if we have time. And that will allow us to do a good review. All right, so it's 7 o'clock. I know we have, some people here. Jamie, I see you. Is there anybody else that's able to see this photo besides Jamie? Or is Jamie the only one?
I can see it.
Okay, thanks. Okay, so let's go ahead and start throwing some terms out. And then what we'll do is try and learn some of this material. All right, so we have the eyeball. And what we're trying to do is figure out, is this the inside of the eye or the outside? And you're going to use number 2 up here. Number 2 is the lacrimal gland. So the lacrimal gland will be lateral above the eye. So this would be the lateral aspect. And this white area is called the sclera, S-C-L-E-R-A. It's a connective tissue structure. And all these extrinsic muscles attached to it. So the sclera that's white here gives shape to the eye. And if we look up in the front where my arrow is, I hope you can see the arrow, that's where we're going to have a cornea. We're going to have the iris or the colored portion, and the pupil is the hole allowing light to get into the eye. So the anterior chamber would be in the front. Obviously, if you have an anterior, you got to have a posterior. Remember how we said that was the way things were? And if we're looking out here, number 3, let's see what it tells us what number 3 is. Number 3, it says superior oblique muscle. Number 4 was the superior rectus muscle. So, basically it's kind of hard for you to tell, that the orientation is not like I would like you to be able to see it. Let's go to the next picture. This gives me a little bit more understanding. At least I can see what's to the middle. So to the middle is this area over here, and on the lateral is the lacrimal gland. This is what's called a superior rectus. So this muscle brings your eyeball, if it were to pull down, your eyeball goes up. And if I have a superior rectus, what did I say? You have to have an inferior. If I got a medial, you have to have a lateral. So this lateral rectus muscle that's right here is the one that ABDUCT abducts the eye, lateral. So it's cranial nerve 6, abducens, recall? Abducens, okay? Number 3 is the cranial nerve that goes to the eye. And it's not the first cranial nerve, it's the what? Second. So it's optic. Optic, see how big it is? All right, so think of the eye. I was thinking it was like an acorn. I don't know why I thought that, but you've got this area in the front, and then the back part of the acorn, the part that attaches it to the tree, is kind of like a brown part, but then there's a little stick part. Well, think of the optic nerve as that, okay? And then these muscles move the eyeball, is what we actually have. This would be like an inferior oblique muscle is what we would have down below there. So let's go and see what they tell us. So if I were studying for the practical, I would go like back and forth and look and just see. So you got sclera, lacrimal gland. They say superior oblique, muscle, but they're all they're all like, we have a superior rectus. Let's go back. So here they're calling it superior rectus. This is looking from the inside out. Let's see where we are. Let's move. This is looking from the outside in. That's superior rectus. That's lateral. This would be inferior underneath. That's the optic nerve in the back. Okay. Let's see where we go from here. So optic nerve was cranial nerve 2. Now we're looking on the inside of the eye. So this would be the front of the eye. This reddish area here is called a ciliary body, and it's made up of a muscle, ciliary muscle, that actually connects to what's going to be the lens that'll be in this opening. This is a choroid. It's either black or a brownish color, but look what it does. See how it follows all the way to the back of the eye? So in other words, it absorbs light and tries to get rid of scatter, is what the choroid, C-H-O-R-O-I-D, does. Ciliary body is this area right in here with a ciliary muscle, and it's going to make aqueous humor, which is fluid in the anterior chamber. This opening would be where the optic nerve would come out. These are blood vessels, but this area all the way around is the retina. This area in the back is called the macula lutea, and it's actually, there's an area of macula lutea in the fovea. So the fovea is the area of highest visual acuity. And we'll talk more about that as we go on. So I think we're going in the wrong direction, aren't we? All right.
I have a question though. Isn't it that when we are looking at a picture that it actually appears upside down in the back of our eye?
Yeah, and your right eye sends a message to the left part. So in other words, your eye is like a large, oh gosh, what am I trying to think of? You know, you've seen these large dishes that they'll point to the heavens. Okay, so think of your right eye as a big dish. And so so what we're going to have is part of the retina of the right eye is looking to the left, is what's going on. So we're sending all these different signals, but it's inverted. So in the back of our brain, basically in the occipital lobe, we have to go and orientate it into the right manner. Okay, but you're correct. Okay. All right, this is the posterior chamber called the vitreous humor area. This is where inside of this you're going to have rods and cones. This is a lens that's going to fit right in that depression. The, let's say, clear area on top here is the cornea, and the hole is the pupil. And then the iris basically controls the amount of light going in. And so with bright, like sunlight, your pupil is very narrow, and at dark, at night, the pupil gets larger. Okay, so we have lens, cornea, the iris colored portion, and the opening is the pupil. Vitreous humor is what would be in this area here. Okay, let's see what else we've got. Actually, I can go over here and I should be able to. Here you can actually see the choroid going all the way to the back. See how it's dark, and that absorbs scatter is what we actually have. Choroid, it's either brown or black. We're going to use that in order to orientate ourselves when we look at this picture. So you see this brown in the right-hand lower corner, that's the choroid. And if you look just to the inside of this, you have little rod-shaped objects called rods, and then you have little cones. So this is the receptor. These are the rods and cones, and they're closest to what? To this dark area. And then the middle part, we're going to have two different layers, and then we're going to have a layer out here. So there's actually three different layers. There's a ganglion layer, a bipolar layer, and then this rods and cones are the photoreceptors. So the light is actually received in here. So let's see where they tell us. So they're saying the ganglion is way out in front. So the ganglion is here. I think I've mentioned a bipolar neuron. So it's a neuron that has like an arm coming off each side. And then these are photoreceptors. Can anybody see these, this long, it looks like a long, like rod? That's how it got its name. So the cones are for color and the rods are for black-white vision. So during the day, cones are primarily used, and at night, rods are used. Okay, does that make sense for everybody? So you're probably going to see, a picture where we're trying to describe, what is, this a picture of? Well, it's a picture of the retina of the eye, and this is the choroid. Does everybody kind of follow? So basically, it says here's the choroid, and then outside would be white. And I didn't see a white outside. I see dark. Maybe that's white outside. That would be the sclera. Okay. All right. So that gives us a little bit of information. That talks a little bit about the eye. Let's go ahead and take a moment while we just did that. Let's go ahead and look at your, let's say your assignment for tonight. It was anatomy of the eye. So page 1, you're supposed to match. Has everybody got it in front of you?
Yes.
Okay, so let's look at aqueous humor. Aqueous humor is K.
Mm-hmm.
And so aqueous humor K, fluid in the anterior part of the eyeball, and it nourishes the lens and the cornea, right? So it's way in the front. The vitreous is L, and it's non-replenished, and it's in the posterior, and it pushes up against the retina and holds the retina in place. The retina can tear and fall away from the back of the eye, and then an ophthalmologist goes in with a laser and kind of burns that or tacks it back to the back of the eye, so you don't have the retina floating off this, off the back of the eye. And I'm sure you've heard of people that had to have, some surgery to have their retina. You had a retina detachment. Well, then they had a retina reattachment. Pupil is G, hole in the center. Fovea, M, right? Area of highest visual acuity, way in the back. So if I'm going to look at something and I want good detail, I want to direct the image that I'm looking at right on the fovea. All right. And after that, we have sclera, which is B, connective tissue, outer white area. Then we have these different muscles on your sheet. Rectus, there's a superior and inferior, a medial, a lateral. And then you have oblique muscles, both superior oblique and inferior oblique. And those are a group of muscles, they move your eyeball. Optic nerve is I. Cranial nerve transmits the stimuli from your eye to what? What part of the brain? Occiput, right? Okay, lacrimal gland is N. That's fluid. So every time you blink, Jamie, and we blink like, a whole bunch of times every minute, we're cleaning the eyeball is what we're doing. You're just taking the dust and stuff that gets on the eye. Iris is F. Colored portion of the eye regulates the amount of light. Choroid D. Notice vascular tissue, it's black or brown, dark, absorbs light and stops everything from refracting or reflecting is what it's doing. Ciliary body, E, secretes aqueous humor, and it has a ciliary muscle. And so the muscle attaches to the lens, and it'll pull on the lens to make the lens either fat or make the lens what? Skinny. But it's trying to direct the image onto the retina is what it's actually trying to do. Ciliary body was E. Cornea, we can transplant corneas, and C is the anterior clear covering. And so because it's curved, it reflects, it says refracts light. So light is being altered as it goes into your eye. And then the lens is J, avascular biconvex structure, and it'll refract light onto the retina. And then the retina is H, and that's the bilayer membrane that has photoreceptors. So that is your eyeball. And so in front of us, we have what? What structure do we all see? The inner part of what? What do we have in front of you on your screen? Anybody?
Looks like an ear.
Looks like an ear to me too.
Mhm.
So the outside ear, if you pull on this is your ear, your earlobe. It's called the pinna. This is an auditory canal, remember? So what area of the skull are we talking about? We're on the side of the head. Where would we be?
Temporal.
Temporal, exactly. And so this is an interesting area. See number 3? What is number 3 doing? It's called the eustachian tubes. Anybody remember? Okay, tell us.
What's it? Well, for pressure and for drainage.
Yeah, so in other words, I had horrible earaches when I was younger, and I would get a lot of pressure in this eustachian tube. You equalize pressure between the back of your nose and throat through this tympanic pharyngeal, canal, but it's called eustachian tube. And so if you're going up in a plane and you hear your ears pop, that's what's actually happening. So you chew gum to do that, or when you swallow, it would allow you to equalize pressure between the ear and the outer nasopharynx or the atmosphere. Is everybody with me? Okay. All right. So pinna is the outer part, external auditory canal was the area right in here. And then this eustachian tube is the area. Equalizes pressure is what it actually does. And here's the eardrum and 2 of the 3 ossicles. So what you can't, it's hard to tell, but this is the tympanic membrane or eardrum. And we have a hammer and an anvil. This looks like an anvil. It looks kind of triangular. This is the malleus, the hammer, and then the incus, and then something called the stapes. But what actually happens, the eardrum vibrates, and then these bones begin to vibrate, do they not? So let's see a little bit more as to what it says there. Tympanic membrane, malleus, hammer, incus is anvil. Let's go back so you can check. So basically we're looking right there. The first one would be the hammer or malleus, and that's the anvil right there.
So then how that works is the process of hearing can be explained by, it's like transduction in a sense.
What you're doing is taking sound waves and trying to create an action potential. Yes. And so what do we know about sound? Sound travels better through what? Metal or water than it does what? Air. So has anyone heard of somebody putting their ear on like the metal railroad track to hear if there's a train coming? You've seen that in a movie, right? Or put your ear against a pipe to, if you hear sound, okay? Even water will, sounds in water are conducted very easily, okay? All right, so good question, Kelly. All right, what do we have here then? This is the part of your kind of like inner ear. Let's go back to the other area. This area in here is considered to be the middle ear. I had a picture and it showed that the outer ear is the pinna, or the outer ear itself, the pinna, all the way up to this area here. And then once you get on the other side, so this is the inside towards the brain side of the ear. It's the middle ear. And so we have a hammer, the malleus, and incus, which is the anvil. And we don't have the stapes until we get where? Right there. See number 2? Stapes means stirrup. So if you're riding horses, you put your foot in the stirrup. And could you get your foot right through that? It's a stirrup shape. So those 3 little ossicles will vibrate. What you might want to know is that if you go to a rock concert, and I don't know if any of you have gone to a rock concert or not, but if you stand real close to the front, what happens to you? What happens to you when you stand in the front? Let's say you're at Raymond James and the Who is going to play, or somebody else.
Don't you get like a ringing in your ear or something like that?
What's gonna happen is it's so loud. There's a tiny muscle that connects to these ossicles that stops it from vibrating. You don't have it on here, but there's a protection that'll stop the vibrations from being too much. But you can still do damage to your ear and to your hearing. Let's go on this other side. This is the cochlea, number 4. The cochlea is for hearing. It means snail in Latin. And then what cranial nerve is number 5? It's cranial nerve what? Cranial nerve, I can tell you it's above 6. So which would it be? It's going to the cochlea and the vestibule. So it's cranial nerve 8, and it's actually 2 nerves joined together. The vestibular portion is going to number 3, and this is where static equilibrium is posture. So this part of the nerve, cranial nerve, is going to the vestibule. And number 1, these are 3 rings like a gyroscope, and those are semicircular canals. That's for dynamic equilibrium, like doing a waltz, wherever you're spinning around. So when you're spinning around, and if any of you has ever seen anybody like down in Al Lang, they used to take the kids and have them spin around with a baseball bat like 20 times and then try to run to first base, and then they'd fall. Has anyone ever seen that? All right, well, if you haven't seen it, it's because when you spin around something 20 times, you really disrupt the little hair cells that are in here, and you don't have any balance at all. So you're going to try and be going this way, and you're going to wind up going over to the side. And people always make fools of themselves when that happens. All right, so let's see what we actually have. Semicircular canals, dynamic balance. Stapes was the stirrup. The vestibule is static equilibrium, just to be able to stand up. Cochlea is for hearing. And then this is the 8th cranial nerve, vestibular cochlear nerve. Is everybody with me? You need to know not only the names of the cranial nerves, you need to know Roman numerals from 1 to 12. All right, this is the cochlear portion, and I'm not too concerned with the scaly, there's scaly tympani and other, but this area, I want to know what that is. So when we look in here, this area above these little hairs is called a tectoral membrane, and it rests on the organ of Corti. This is for hearing right in here. So in other words, when we look, what we're going to find is the vibrations in here, this little membrane sits on top, and an action potential is generated. And then where's the nerve? That's the nerve right here. Does it follow where we're coming? So from the cochlea, this is the cochlear portion of the 8th cranial nerve. See this right in here on the left? Does everybody see me moving the arrow? That's I'm going from where the organ of Corti is to the inside right there. Can everybody see, or am I talking to myself?
I see it.
Okay, so that's the cochlear portion right there. Okay, so you need to know that this is the organ of Corti. All right, let's go on. All right, so they go into scala vestibuli and scala tympani. You want to know vestibular membrane and organ of Corti. So the organ of Corti has this membrane on top with what? Little tiny hair cells. And this is for hearing. So number 3 and 4, you need to know that. Okay, what do I have here? Jamie, you have any clue what I've got there besides the slide?
No.
Okay, stick your tongue out. Go ahead. All right. That's your tongue, girl.
Okay.
Okay. So for everybody that's with me, what we have here's the top of the tongue. The muscles are down here, but these are little crevices. And so these are gustatory cells. They look like little tiny circles, and there's little like hairs that come out. So food gets in here, and that's what allows us to taste. So it's a chemical reaction that actually occurs. So if you notice, even up in here, there's another one. So the receptors for taste are in your tongue and at the back of your tongue. Let me go turn my cell phone off. Sorry. Okay, very good. We're back now. At least I am. Sorry for that little delay there. All right, so looking at what we have here, this is a slide of the tongue, and we're talking about taste. And what we're looking at are the receptors for taste. And you, in your wisdom, know that cranial nerves 7, facial, 9, right?
Mm-hmm.
And 10, vagus, are all for taste. You know that, right? 7, 9, and 10, because you memorized it. And then Dr. Byrne told you 7, 9, and 10 for taste. Okay, let's go on. We may be at the end. Oh, there's taste bud. Okay, so basically, facial nerve, glossopharyngeal is 9, and also the vagus, and they're all going for these are little taste buds, and they're going to the parietal lobe. Taste is parietal lobe. Okay, so I'm going to stop sharing, and then we're going to go ahead and you'll be able to look at me, and we'll see who's here in the class. So hopefully that'll stop. It'll take a moment, and we'll be able to get everybody together. Okay, good. All right, so looking at these images is going to help everybody in order to do well on the practical that's coming up. And so I have an eye in front of me. Why don't we use it, and let's talk about some of the stuff that we just learned. Can everybody see my little eyeball? Okay, so the white outer part is called the
Sclera.
what? Sclera. And then these are extrinsic muscles on top. So superior rectus, and what I'm looking at here, take my word for it, this would be the lateral rectus. So with the lateral rectus, if I can move my eyeball laterally, I'm abducting my eyeball. So I may have a question that this one, the lateral rectus note, is abducens. Everybody follow?
Mm-hmm.
Trinity, you got me? Okay, this one on top is superior rectus. And so if I have a ball and I pull it backwards, I can look up. That's ocular motor, that's cranial nerve 3. And remember I said that there was a poli or a trochlear nerve, cranial nerve 4? Well, this superior, this is an attachment, a tendon. This would be going to the superior oblique, and it would be the trochlear nerve going to that muscle. Does everybody kind of follow? So cranial nerves 3, 4, and 6 are eyeball movement. Okay, all right. So let's take a look. The whole is the pupil. What's the colored part? Your what? Iris. And it will become more narrow and make the pupil narrow or large. So let's pop this off. And so what I'm going to do is open it up, but I scotch taped it too. Okay, so we're going to open up the eyeball. And so I have a posterior chamber. Does everybody see this post? That's where the vitreous humor would be. And then in the anterior chamber, I said anterior to this structure. What was this structure? It's a little tiny, it's clear.
Lens?
Yeah, it's actually the lens. And there was something attached to it that would pull it, that would change its shape. So it was the ciliary body, and there are little ciliary muscles. So in other words, we're trying to direct light right onto the retina. So you notice a few of us have glasses, and we wear glasses because without them, we're not really getting an image on the retina. So these lenses have a shape, and they're trying to get it on, but I can go and squint. You see people squinting. What do you think they're doing to their lens? They're changing the shape of the lens, trying to do what? Get the image on the retina. So anything anterior to this is in the anterior chamber, where you have aqueous humor, and that's replenished is what we have, and that's made in the ciliary body. And if I look inside, there's a choroid, that's a dark layer. okay, is what we actually have. So that gives you some understanding about the eye. What we want to do is, let's talk a little bit about page 2. And what do we have on page 2? On page 2 of your assignment that we have for tonight, it says 3 layers of the eye, right? And it says sclera on the top left. It gives what to the eye? Shape. And the choroid is the second layer. It's absorbing light. And then what does the retina do? That's another layer. That's where the photoreceptors are located, right? Okay. So in the retina, you've got 3 different layers. Remember I said the photoreceptors are rods and cones? So I'm going to read you what I wrote down because I went and did this. And how did I get this information? I went to the notes from the lab. And so I'm just transferred it over is what I did. But the photoreceptors are going to, basically, it's a graded potential, but we're converting what? A light impulse to a graded potential in the photoreceptor layer. That's all we're doing. We're trying to get an action potential is what we're actually doing, okay? And the thing you need to know, cones, C for color, R for black and white, right? Just remember C for color and you'll be fine. So the bipolar cells carry the vision information from the photoreceptor. So the receptors are way in the back, right? They're trying to convert these light impulses into some graded potential. And then that message goes through the bipolar, and then it goes to the ganglion cell. And the ganglion cell creates the action potential and takes it to the optic nerve. I just think you need to have some understanding. So there's 3 different layers, but the ones that we're going to be the most, let's say, interested in are rods and cones. Okay, now for lecture, they may want you to know, what the ganglion does and what the bipolar does. All right, let's go over the different matching for the ear. That's on page 2. So auricle or pinna is what? G?
Yes.
Funnel sound. The auditory canal, external auditory canal, D is what? It creates wax, but it also funnels sound to the eardrum, does it not? Okay, what's wax do? Why do you have wax? Anybody?
I think it's also so that things don't fly in your ears. Like does create like a smell and kind of a like antibacterial in a way.
Yeah, it is, and it'll also trap bugs. Is what it's supposed to do. Okay, and then next one is the tympanic membrane, which is your eardrum. And let's let Justin into class. Okay, so tympanic membrane is H, and so H converts sound waves to vibration. Okay, is what we have. And then remember, you got ossicles, hammer, anvil, and stirrup. And so there's C. They conduct vibrations through what part of the ear? It's the middle ear. Hi, Justin. So we're on page 2 is where we are. And we're just talking a little bit about the ear right now. And then we said the cochlea is A. That's for hearing, right? The spiral canal contains the hearing receptors. And I already forgot what the name of the hearing receptors was, Jamie, Trinity, Seth, Jennifer, Nicole, Kelly. What was it? Organ of who? Organ of Corti. Remember? And organ of Corti is B, hearing receptors. And cochlea was A. And the hair cells of the organ of Corti is B. And the vestibular cochlear nerve, I forgot what cranial nerve. It's not 5, it's not 7, it's not 9, it's 8, 8th cranial nerve. So I think I've only done this class 100 times over the last 20 years, Kelly, longer than some of you are even walking on this planet. So you got to know your cranial nerves, Jamie. If you want to do well on the test, cranial nerves is where it's at. I must have 40 questions on cranial nerves. Okay, out of like 100, but you're only going to get 50 of them. Okay, and then vestibule is I. So let me turn that off. Sorry. So sorry. All right. So semicircular canals are J. That's for dynamic equilibrium, J. Dynamic equilibrium is dancing so you don't fall on your face. Okay. And static equilibrium was the vestibule. So the 8th cranial nerve is for balance. That's the vestibule. And the semicircular canal. And then the cochlear part is for hearing. Is everybody with me?
Mm-hmm.
Okay. All right. On page 3, they go into, we already talked a little bit about the organ of Corti, the hair cells, and the vestibular cochlear nerve. That's at the top. We looked at a slide of the tongue when we were looking at it, and you need to know that there were basically taste buds. Taste buds are those little gustatory cells. Gustatory means taste. And I said cranial nerve 7, 9, and 10 were for taste sensation. And so we have a lot of different receptors that are in the body, and some of them use chemicals, like for smell or for taste. Some of them use light, like the rods and cones, but they have them listed. So you have 5 special senses. What are they when you look at this sheet here? What are the 5 special senses that are on the sheet?
Sight.
Sight is vision.
Vision, hearing.
Hearing is 2. That's another one.
Equilibrium.
Equilibrium is balance. That's 3. What is this here?
Smell.
Smell is there. I think we got them all, didn't we?
Well, taste, yeah.
Taste. So that's all 5. So those are special. So what did we say? We said taste, hearing, sight, equilibrium,
Smell.
right?
Mm-hmm.
Smell. Those are special senses. They're special receptors. Now there's others that are general receptors. So a general receptor is not special. It's not unique. You kind of understand? So in other words, remember we had a free nerve ending. Free nerve endings were for cold, temperature, or pain. Remember nociceptor, okay? And we had, in the skin, we had 2 receptors. One was called Meissner and the other was Pacinian. So Meissner is like light pressure and Pacinian is deep pressure or pain. So if I have something just barely walking across the hairs, let's say on my skin, Meissner would pick that up because it's light touch. But if I have something pushing down hard, then you're not only pushing through Meissner, now you're pushing into Pacinian. So you need to know, is it a general receptor or is it a, special? Does that make sense?
Mm-hmm.
Okay, so let's make sure you understand what type of receptor it is. Light is photoreceptor. Just put a P there on the bottom of page 3, right? So light is P. Smell is a chemical. C, put C. Equilibrium is mechanical. So in other words, when we start talking about in the vestibule and in the semicircular canals, there's like little hairs and there's little stones that are going to fall on these hairs. So Kelly, when I turn my head to the side, I know I've turned my head to the side because those little stones are moving in my semicircular canal and sending signals to my brain, letting me know that, I've turned, torqued my head to the side, okay? And so it's a mechanical, it's like pressure, that's mechanical. Pain is N for nociceptor. So a pain receptor is a nociceptor. Taste is chemical. Pain or pressure is mechanical, put M. And if I touch somebody or touch, that's mechanical because I'm pressing. And then even hearing, remember I said you have the little hair cells or the organ of Corti, and the tectoral membrane is pushing on top of it, correct? All right. So that gives us a little bit of information, but we now have page 4. And so as we go on, let's look at page 4. Page 4 wants us to know what's the sense and what kind of receptor is it? And then what cranial nerve? I know you love cranial nerves. And then what part of the brain, what brain structure or what lobe? So let's take the first one. We'll go straight across. And let's talk about taste. Taste, cranial nerve 7, 9, and 10. It's a chemical for C. And cranial nerves, what did I say? 7, 9, and 10. So what part of the cortex? It's going to the primary taste cortex, is called the primary gustatory, gustation, and it's in the P, parietal. So just put P, parietal, and primary gustatory. Okay, so everybody got that one? Okay, so let's do smell. All right, so smell is chemical receptor, and it's the first cranial nerve, olfactory. And what part of the, let's say the brain, the primary, put 1 with a little dot on it. That means primary olfactory area, and it's in the temporal lobe, temporal, T for temporal. And that was in the primary olfactory area. Next one is vision. Vision is a photoreceptor, P, just put P. And we already did olfactory, which is cranial nerve 1. We said the optic nerve was what?
2.
Eyesight? 2. So 2 is vision, optic nerve, and it's going to go to the primary visual area in the what? Occiput. Put O for occiput, occipital lobe. Is everybody with me thus far? Okay. And hearing. Hearing is a mechanical, put M. Hearing is what nerve? 8th cranial nerve, called the vestibular cochlear nerve. Yeah, you're getting it now. Okay. And it's the primary auditory lobe area in the temporal. So my ear is on the side of my head, and so I hear on the side of my head. My vision is different, right? I have my eyeball in the front, and yet I see with the back. That always amazed me. My occipital lobes are where I see, okay? So vision is occipital lobe. Hearing is the next one, and it's a mechanical vibration, and it's the 8th cranial nerve. It's in the primary auditory, or the hearing center, and it's in the temporal lobe, is what we have. And so let's go ahead and look at equilibrium. Equilibrium is an M for a mechanical, and equilibrium is the 8th cranial nerve. And with equilibrium, I need balance. So I need my cerebellum.
Mhm.
Cerebellum has to help me to keep my balance. Okay, so that's the part of the brain. But on the cortex, am I going to go to the primary sensory or primary motor, because I'm dealing with equilibrium? I'm going to be going to the primary motor, and motor is before, motor is in the frontal. So remember when we were looking at the brain, I've got a brain here. So we got a brain. So we said the frontal lobe, it was motor, and then we have a gyrus, and then we have the parietal lobe, it's sensory. M is before S, correct?
Mhm.
And so on the side, temporal for hearing. In the back, occipital was for vision. Okay. And so you would love this to be on your test. It actually gives you answers. Justin, it tells you that hearing is in the temporal lobe. You would like that during the test, I know. I would too. Okay. Okay, pain. Where am I gonna, it's a nociceptor receptor. And so there's different areas. If I'm talking about the face, it's a cranial nerve 5. Cranial nerve 5 is sensory to the face. Remember somebody got slapped in the face, Kelly? And they had an ophthalmic, a maxillary, and a mandibular. And when they got slapped, they had 3 marks on each side. Remember, they had 3 marks on each side. And so trigeminal is the 5th cranial nerve, and it goes to the face. But what about in the rest of the body? The rest of the body is going to be sensory nerves coming from the skin, right? Of my arms and legs. So in other words, the lobe or the area is going to be the sensory somatosensory area. So it's primary SS, put somato means body, sensory. And remember, what was I saying here? Motor was in the frontal lobe, motor's in the front, parietal is sensory. So parietal lobe is where I have sensation. So if somebody sticks you with a knife or you cut your finger, you feel that in the parietal lobe. And then we're almost done. We got one more. Touch. If I touch somebody's face, okay? It's a mechanical, M for mechanical receptor. If I'm talking about the face, it's the what? Basically the 5th cranial nerve, number 5, trigeminal. If we're talking about down here, it would just be a body somatosensory nerve. So it would go to the primary somatosensory part of my cortex, which would be parietal. Okay, is what we would actually have. All right. I think if we're able to, I'd like to go and talk a little bit about what is in store over the next couple weeks. So we've just done the 3rd lab for this test that's coming up. And you're going to take a quiz over special senses, which will be lab 12. I think what we need to do is see if I can share screen with you the images from labs 10 and 11. You want to do that now for like the next 15 minutes? And that'll get you ready as a little bit of review for the test that'll be the end of the month. So what I just did is I opened the final practical 3 on the Sunday at the end of the month. So Sunday, I think, is the 29th. I opened it up. So all day Sunday, it's available. The Monday lab, it's open all day Monday, and your Tuesday lab. So it's open all till Tuesday night at 11:30. And then I have a few people that'll be taking it after that. But that gives you 2 full weeks to be studying. Is everybody kind of with me? Right? So what I would like to do right now is, unless you have a question, does anybody have any questions thus far? You would, go ahead.
I have a question. It's not on the schedule. We keep going through Thanksgiving, correct?
We're not. I just took off the Zoom for, I left 2 Zoom classes, but I'm trying to cover with you what I would have covered next week, because normally they don't have, let's say, lab classes. You get the week off typically. So you understand?
So next week we have the week off.
You have the week off. Well, you better be studying, girl.
Okay.
But you don't have to listen to me or look at my face. But that's only if I can get through these 2 labs that we we want to go over lab 10 and 11 and explain a little bit so it makes more sense to you guys.
Okay.
Is everybody okay on that? All right. Yeah. So I think we kind of introduced the special senses part. So what I'm going to do right now is see if we can share screen, and then we'll go from there. And it looks like I can get to, let's do lab 10 first. Let me see if I can get it here. All right, it's going to take a little time, so bear with me as this comes up on the screen. Can you guys see this?
No.
No. No, sir.
You can't see it? Ooh,
No.
Okay. That's not going to help, is it? because I can see it. All right, I'm going to talk about lab 10 and you're going to listen. Is that okay? Is that going to, that'll help a little bit. Let me try a different approach. So what do you see? Just a blank screen right now?
Yeah. Just all of our faces. Did you hit shared screen again in there?
Okay, I thought I did. So let me go back down because let me get this down because it says share on it. I thought I hit share screen. Let's go. Let me do it this way. That's 12. And let me see, there's 11. Let's go to this one. Okay, can you see this now?
It's, yes.
All right, I'm gonna hit it twice and it should come up, it'll get larger. Is everybody with me?
Yep.
Okay, so Nicole, you can see now?
Yep.
Okay, all right. Thanks on the share screen again, so that helped. All right, what do we have here? This is the spinal cord, and it basically, you have layers. So let's just talk about the layers. These are what are called meninges or layers. Number 1 is the dura, D-U-R-A. Number 1.5, you don't see 1.5, do you? Well, the gray part there, just underneath the dura, is called the arachnoid. Is everybody with me? And so
We're only seeing thumbnails. I'm only seeing thumbnails.
I got it larger. You don't see anything bigger than?
No, we just see it in
Oh, like a little tiny thumbnail.
your folder.
Mhm.
All right. Well, you know what I'll try and let me see if I can do something different. Let me stop share. Okay. Because the whole idea, let me get rid of that. What I'm going to go ahead and do is let's see if I can try a different approach. And all you see, you don't see like a large screen, you just see small.
Yeah.
All right, that's not going to work. I think what we're going to go ahead.
I saw the large screen for like half a minute, like a
Yeah.
quarter of a minute. So it kind of like, did you push a certain
What I did
button?
is I hit it, I basically just made it, I double clicked and then it got larger. Can you see now?
So what I'm wondering is maybe if you minimize where you have the little, the smaller pictures, minimize that screen, maybe we'll be able to see the larger picture behind it or something. Because I seen it for half a second and then it disappeared.
Okay, let's see if we can work on that. Let me do this.
Do you want me to try?
Yeah, Nicole, can you do it, you know, in one of the other, because I think it's set where if you can show it and it comes up, I'll talk, because that's what we need to do. You want to give it a whirl? It doesn't matter. Go to lab either 10 or 11, okay?
I'm in 12, I think.
Yeah, that we were just in 12.
Is this what you want?
Yes. See, now all I see is small, but let's talk a little bit about it. I don't see it as large. Can you double click it and see if it gets larger?
That's as big as I can.
Okay, well, all right, let's talk about what's there and what I see. This is a cross section of the spinal cord. And what you have there are three different layers. There's a dura, the outer, and then underneath it, arachnoid layer it gets its name because of, it looks like a spider. And then the subarachnoid space is where there's cerebral spinal fluid. And then right on the spinal cord is called the pia layer. Okay, so the pia layer is intimately on the spinal cord. When you look at the spinal cord itself, what you're going to see are sensory nerves going into the spinal cord. And they're sensory rootlets, actually. So the sensory rootlet that goes in is actually the part of the sensory neuron that's the dendrite, okay? You need to know that there's a dorsal root ganglion, and that's the group of sensory cell bodies, and that's outside the spinal cord itself. And then you have the butterfly. So when you look at the dorsal horn, going into the dorsal horn is the dendrite of the sensory nerve. And then between that and the motor nerve is actually an inter or association neuron. So the dorsal horn is going to have the cell body of the association or interneuron. The ventral horn will have the cell body of the multipolar neuron, and coming out of the spinal cord is actually the axon, is what you actually have. You need to know central canal, and that's the little area in between. It's the isthmus between each side of the spinal cord. If you want to change the picture, go ahead and do that now. Are you able to change the picture?
I hope so.
Okay, if you can't, okay. So this is just basically what you were showing up there are some of these terms that you need to know. So I have the model right in front of me. You need to know dorsal root ganglion. It's the swelling and its sensory cell bodies. You need to know sensory root, and it's actually the axon. The axon is, I say dendrite, but it's actually the axon of the sensory nerve. And then you have a dorsal horn and a ventral horn. So dorsal horn is sensory, and anterior or ventral horn is motor. And so the motor axon is going to go to the effector, which happens to be a muscle or a gland. Go to the next slide.
This one, or go to another one?
Yeah, just go to another one. All right, so we're still in the spinal cord. And so basically, let's talk about a reflex arc. Reflex arc has 5 parts to it: sensory receptor, sensory neuron called a unipolar neuron, an interneuron or association neuron, and a motor neuron, and an effector. Go to the next slide. And go to the next slide, because that's hard to read. I can't even see. Okay. Okay, they're just showing different parts of the cross section of the spinal cord itself. And let's go to the next slide. And so they're asking different questions. I ask questions like these same questions. You know, like, what is the dorsal root ganglion? Identify it. What number would that be? You know, so there'll be questions like that. You need to be able to tell dorsal horn from ventral horn. And the anterior fissure tells you which is the anterior and which is the dorsal. Go on, next one. Okay, this is a nice slide, because remember the different layers of nerves? Well, we had different layers of muscle. So these are fascicles, and so perineurium is around each of these 3 little fascicles. Around each individual nerve is an endoneurium, and around all 3 of these is the outer, is called the epineurium. So you need to know epineurium, perineurium, and endo. and perineurium is around the fascicle. So each of these groups of nerves, it's a cross-section of axons. Go to the next one. And it's just showing what I talked about. And what do you have for the next? Okay, it's kind of hard to tell. I can't even tell what that is. Go to the next slide. Okay, well, I think it was just, you know what, if you go back one, all it's showing is the individual axons, and they have Indian ink. It's a cross section, and it's the endoneurium around each axon. Okay, is what we have. Go to the next one. And it's just showing, explaining endoneurium, perineurium. And I'm not sure what that is.
This one says cranial nerve that it's showing on the.
What's it actually? Cranial nerve, which one going to what?
It's on the eye, so it would be 2.
Yeah, if it's the optic nerve, if it's an eyeball, then it would be optic. Okay, go to the next one.
Hold on, takes a minute.
It sure does, doesn't it? Okay, and I
guess it was, I don't know, it says, I guess it was the trochlear, abductus, and.
Okay, so abducens. So let's go ahead. I think we're going to get through this, and then I've got some models here, so we'll talk about some stuff, and I'll just show them to you. Let's go through the rest of this. Okay, so this is the eye, and we've already looked at these. So in other words, these were I was able to show earlier, is what I was able to go ahead and do. So what I'm going to do right now is see if I can get back. I'm going to let you stop sharing, and I'm going to see if we can all get together. Okay, so I'll try that. So I see you, Nicole. I'm going to see if I can minimize this and return the meeting, and we're all together. So what I have in front of me are some of the things that we just talked about, because I need to get rid of them. Okay, so remember when we looked at lab 10, we were looking at different parts of the nerve itself, and we said that that these were the dendrites going into the cell body, and that this was the axon hillock, and this would be the axon. Does everybody kind of follow? Okay, so dendrites are graded potential going in to where the action potential occurs, where the axon hillock through the axon. This is an axon, but there's indentations where there's no myelin, right? So we called that node of Ranvier. And we said that saltatory conduction jumped from one node of Ranvier to the next, remember? And it was a real rapid type of conduction. Do you see what we got? And we said that the neuron would be this area right here, but wrapped around it was the what? Schwann cell. So the Schwann cell is this white. And so the purpose of the Schwann cell is to insulate this action potential. And so that's what we have here. Node of Ranvier is here, and this is an axon. So if you look on the end, you see how you see the axon with an endoneurium. So in other words, it's like my pencil that I have. You know, think of this as the outside of it. The lead is the axon, and the wood would be the endoneurium. Does that make sense, Justin? You see what we're talking about? So that'll give you some information. So a model like this, you want to know these different parts, okay, is what you need to know. Myelin sheath, and, you know, basically dendrites. This is the cell body. This is the axon hillock, node of Ranvier. These white things are Schwann cells, endoneurium. And so basically that's what we actually have. And so where's the axon going? It's going to an axon terminal, and then the axon terminal is going to go to an effector. Okay. All right, let's take a look at this one. This is a reflex board. And so it's basically doing the five parts of the reflex arc: sensory receptor, sensory neuron. What's that? Interneuron or association neuron. What is that? It's going to a muscle. Motor, what? Neuron. Sensory neuron is unipolar. And so basically the sensory neuron, node of Ranvier. So if you're thinking about it, what goes towards the cell body? Dendrites. What goes away from the cell body?
The axon.
Axon. So this is a sensory, a group. Let's start with this. Let's say 100, 500 of these cell bodies are put together in a dorsal root ganglion that's outside the spinal cord. This is what becomes part of the sensory rootlet going into the spinal cord. This is the association neuron. This is in the dorsal horn, this interneuron. This motor cell nucleus is in the anterior horn, and I've got an anterior horn. Coming out of the spinal cord is a motor rootlet, and so this is going to an effector. Does that make sense?
Mhm.
All right. So Nicole was gracious to put this up. Remember, that's what you were looking at. And so what were we actually looking at? Well, we're looking at a spinal cord. How do I know which is dorsal? Well, I know it's dorsal because this is a this is a cervical vertebra. See, it's bifid.
Mhm.
And so these are facets. Here is the what? Here is the spinal cord itself.
Mhm.
So this is the top part is dorsal and the bottom part is ventral. I know that because there's the anterior fissure right there. Does
Mhm.
everybody see the anterior fissure? All right, what goes into the dorsal horn? These little fibers are sensory basically, this is the sensory axon going in here. In the dorsal horn, I have the association neuron that's in this part. What's down below? The motor neuron nucleus is down in the anterior horn, and then coming out is the axon or motor rootlet, and that's going out to the body. In the outside, there's a group of a fat area where I have sensory cell bodies. That's called the dorsal root ganglion. Okay. So in other words, I'm going to ask questions. This is the dorsal root ganglion. This is the dorsal root. That's the dorsal horn. That's the anterior horn. That's the motor rootlet. Does everybody kind of follow? So that's the central canal, and then there's fluid around it. So that'll be something that you'll want to know. Okay, I think we're at a good point to stop for right now. And I think we've covered about an hour and 10 minutes of material. Okay. The way to study for the test: look at the image, go back and forth. It'll give you what they are. You need to know two things about the image: what it is and what it does. Okay, you need to be able to identify it. How do you know what do you need to know? Kelly, how do you need to know what you need to know? Look in your let's say the lecture material for the lab. So when I'm looking under hearing, it says hearing in equilibrium. It says receptors, hair cells in organ of Corti in the ear. So you need to know organ of Corti, and you're going to see that in the image. Outer ear, you need to know pinna. You need to know the external auditory canal. You need to know tympanic membrane. So you need to know those terms. So I would read it 3 to 5 times, each time gaining information. I could ask what makes up the middle ear. It has it together. Middle ear are the ossicles and the eustachian tube. That's the middle ear. What makes up the inner ear? The vestibule, the semicircular canals, and the cochlea, they're all inner ear. So see how it's organized. So you just kind of break it down. It's not easy, but you kind of get it organized and then look at the images and go back and forth. Okay. You'll have a lot of opportunity to retake these 3 quizzes. And then I put on the calendar the new time now that the practical is open. And so if anybody has any questions, you want to stay online with me or send me an email, during the next week. But you have 2 weeks basically to get ready for this next practical. And I think with the material, it's only 3 labs, but there's a lot of material. How do we grade you? You're graded on what? 3 practicals and all the quizzes, and there'll be 12 quizzes. So let's say you don't take a quiz, I drop 2. But I mean, if you're gonna, all that's on this next test is what's in labs 10, 11, and 12. So you wanna put your energy there, is where you wanna be, okay?
You have on the Zoom, and I'm not too sure when, you have us after the 30th going all the way to the 15th.
I got rid of that. I got rid of it tonight.
Oh, you did? Okay,
Yeah,
so when officially is like our last class day?
The last class day, as far as us meeting, it's gonna be tonight, because we're not gonna meet next week, okay? You can contact me. I kept the 2 Zooms, but I'm not sure we're gonna use them, because I think with the information that we've already covered, everything's there. You don't need me to tell you this is in other words, what I was trying to do from lab 10 is this is a multipolar neuron. Well, that's a motor neuron. There's stuff in those slides, just know those slides inside and out. And if you do that, you're gonna have an hour, probably about an hour and 5 minutes to take the test, and it's matching, and you're able to go back, but you can't right click, no right clicking, but you can go back and forth on it, like questions, let's say you want to go back and check your answers, but it'll submit after like 65 minutes is what it'll do. But I need somebody to take the test on Sunday. I want to make sure that it's open and working. That's why I opened it early. Does everybody follow? So at the end of the month, I think it's the 29th, I opened it up for that Sunday. And typically, I think I had only 3 people out of like 40 that even took it. I had 30 people take it Tuesday night at like 10 o'clock, and it closed at 11:30. That's not good, Jamie. Okay. Any questions? Any more questions?
Are you talking about December 28th?
I'm talking about the end of November. So the last Sunday of November, and the Monday and Tuesday, it closes December 1st. So I think it's November 29th, don't hold me to this, and 30th, and then it closes December 1st at 11:30. I set that up tonight. So when I set up the labs, I didn't really know, when we end. Grades are due December 11th. Lab grades always go in, not always go in, but we get done with the practical the week before lecture does. So in other words, we always do lecture and get that, we always do the lab and get that done the week before you have your lecture test, okay? And the reason it's set up is normally I have to grade it by hand, but now that I have it as multiple choice, it's a little easier. But we need to, be working on, labs 10, 11, and 12, and then just get yourself ready to take the test, and you don't, you need to go ahead and put the kids to bed, send the husband away, feed the dog, put the dog outside, all those things, right? Does everybody understand? Okay, any other questions that we have? Okay, if not, I think you guys have a handle on it. Know your cranial nerves from 12 to 1 and 1 to 12. Know Roman numerals, know the name, know, what they do, and stuff like that. And that'll help you quite a bit. But it's using those photos and then, what is it? What does it do? So A and P, just like it says, anatomy and physiology. Okay. All right. Catch you later, guys. If you have any questions, hang out with me. Okay. And thanks for helping I enjoyed being with you tonight.
You're welcome. So it's 50 questions, right?
Yes, ma'am.
It's still 50 questions, and you said it's matching. So
Matching, yeah, true, false, and multiple choice. So there's no fill in the blanks.
Okay. Okay.
But you still need to know it. So if you look at your notes, you know, what's part of the middle ear? Ossicles and eustachian tube. What is part of the inner part? You know, you need to know vestibule and semicircular canal. And I told you that, you know, organ of Corti and the hair cells, those are important. And I cannot tell you enough, Trinity, know your cranial nerves, girl. Okay. So if you do that, and you're not supposed to have paper open, you realize that everything should be closed when you take the test. And so you want to be honest with yourself, but just you want to know the material, and you're going to use it later in your careers,
right?
As you get into, you know, if you're going to nursing or into the health field anyway. So you just want to kind of be able to do that. Okay.
But even if we were in lab, like we would have still had like a piece of paper to be able to write like the mnemonic for the
That's true. Yeah, you can take just a blank sheet of paper and do a mnemonic if you wanted. I've allowed people, when we were doing it in lab, they would go 1 through 12 and write the names down and just kind of help themselves. But you don't have time to go looking through, and you're not supposed to be doing that anyway. But the cranial nerves, they get to be fun after a while. So, you know, and you're going to learn more and more and more as time goes on. And if you think this is hard, this is not hard. This is easy as compared to what you're going to be asked to learn. My son went to DO school, and on one test, they had 950 slides for one test for pathology. And I mean, they just throw stuff at you. You either get it or, you know, or you can't get it and you're not there long, Jamie. So you got to get it. It's what you got to do. So the key is just study a little every day and study effectively. Okay, that's the key. I'm preaching to the choir. You're my better students here, guys. So you keep working. So just keep trying. And don't, you know, everybody makes a, you know, we don't do well all the time. You can't. Okay. So have a safe Turkey Day. Don't have 10, 20 people over at your house. Tell them to stay home. Tell them you got to study anatomy. All right. Okay. I'll catch you later.
Thank you.
All right.
Okay.
Bye-bye.
Thank you.